So in this lecture, we will see the different physical tests for the analysis of water. There are different sources by which water is physically contaminated. So we will see each of the different, we have already seen the different parameters by which physical contamination occurs. Now we will see how to measure these parameters. So before moving on to the tests, we have to first see what are the different precautions to be taken before the analysis of water. So we have to test water. For that, we have to collect a particular sample. Okay, a representative sample should be collected. How could we collect that sample? What are the precautions to be taken while collecting the sample? So these are the things which are given here. So water sample should be collected in white glass bottle having well fitted stoppers. It should be of a white glass bottle having well fitted stoppers, and the bottle should be thoroughly cleaned. And if we are taking water from pipe, then we have to allow the water to go waste for at least 2 minutes from the pipe, because some impurities may there in the pipe. So we have to allow the water to go waste for at least 2 minutes, and after that only we have to collect the water. And if we are taking water for bacteriological test from the pipe, then we have to first flame the pipe, flame the pipe to hot, and after that we have cooled the water using the running water, and then we have to collect the sample. So these are some of the things. And another thing is, when we are collecting water from an open source, we have to first remove the stopper and we have to insert the entire bottle into the water. Okay, so using a clean string only after suspending the whole bottle under the surface of the water. So you shouldn't remove the stopper before collecting the sample. So that is the thing, I'm sorry. So you shouldn't remove the stopper before collecting the sample. So you have to first dip the entire bottle into the water, and after that you can remove the stopper by means of some string and all. So that is the thing that you have to note. Now bottle should be held as far as away from its neck position. So we have to handle the bottle before below the neck position. So if there are any impurities in our hand, it should not be mixed with the sample. And after sample collection, this is the important point you have to note. The stopper of the bottle should be well secured, and bottle is labeled with source date and time of collection. So these are the main things you have to note before taking sample from the source. Now, there are mainly three tests: physical, chemical, bacteriological. So, all the three pollutions are there. So, for testing the three pollutions, we have three types of tests. So, in this lecture, we'll move on to the physical tests. There are mainly four parameters. Mainly, four physical parameters are there: color, taste and odor, temperature, and turbidity. And some other physical parameters are also there: density, electrical conductivity, radioactivity, viscosity. So all these parameters except turbidity we will discuss in this lecture. First thing is color. Color can be explained in two main terms. One is apparent color and the true color. Apparent color is the color which is developed by means of suspended matter in water, while true color is developed by means of dissolved matter in water. So if you want to find the true color, we have to first centrifuge the sample. Sample should be centrifuged, and that uh, after centrifugation, what will happen? The sediments will be settled down. Okay, the supernatant clear liquid will be there above the sediments, so that liquid have to be taken for testing true color of the sample. Okay, that is the thing. And there are many sources by which water is getting colored: algae, degraded organic matter, wastewater from industries, divergent species, etc. are some of the main sources by which water gets color. Mainly this color is measured by using platinum cobalt method. So it is a visual comparison method. We will compare the color of our sample water with some standard colored water. So that is the thing that we are doing. So for that we will use the platinum cobalt solution. So we will mix this platinum cobalt solution with some standard sample, or some distilled water or something, and the absorption of light by this standard water will be already measured. Okay, so that value we know. Now we will take the sample and do the same with our sample, and we will compare the absorption of colors of the standard solution as well as this sample. So that is the thing that we are doing, and we will collect this samples, we will compare the samples with non-concentration of colored solution. Special properly calibrated glass color discs or tubes called Nessler's tubes are used. Okay. So Nessler's tubes are the tubes inside which this water is kept. The standard water as well as the sample is kept in two tubes. They may be known as Nessler's tubes. And the platinum cobalt scale is introduced by a scientist known as Alan Hazen in 1892, and it detects specifically, this method is particularly used for yellow color and common method of comparison of the intensity of yellow tinted sample. So the procedure for this method is given by ASTM. So we will see what are the procedure. It is a standard method. This platinum cobalt method is a very standard method, and the unit of color by this method is that produced by 1 milligram platinum per liter of distilled water in the form of chloroplatinate ion. So a solution will be there. So the solution will produce some color when light falls on it. Okay, that color is the standard for comparing all other samples. So that is the thing. The cobalt to platinum ratio will be varied according to each mixture, and the permissible limit of color for domestic water supplies is 220 ppm. So we cannot use this method for highly colored waters. We can use only for some domestic sewages and all, we can use this method, because for highly colored water we cannot use because it detects mainly yellow color. Other than that it cannot. Okay. And this is the way by which we will form that standard solution. So we have already said that our sample is compared with some standard solution which is made up of platinum and cobalt. So this is the solution. This is how the solution is made. We'll take 1.245 gram of potassium chloroplatinate and mix it with cobalt chloride, 1 gram of cobalt chloride, and again we will mix it with water. So these three are mixed together, and we will carefully add some hydrochloric acid with a specific gravity 1.19 into the solution, and then we will dilute the water to 1 liter. And for this solution, the absorbance of 500 platinum cobalt stock solution in a cell having 10 millimeter light path is taken as the reference for color of water samples. So for this solution is kept in a tube known as Nessler's tube. This type of tubes are there. So in this tube, this solution is kept, and we will allow the light to pass through a 10 millimeter light path. A 10 millimeter diameter light path will be there. So we will allow the light to pass through it, and we will observe the absorbance of that light in this solution, and that will be the standard for testing the samples. This standard solution can be used in Nessler's tube, Tintometer, an instrument is there, spectrophotometer, this is a very recent instrument which is used to measure color. So for all these instruments we can use this standard solution. Okay. Now color measurement by using tintometer. Tintometer is another an instrument which uses the standard solution for measuring color. It has two holes and an eyepiece. Eyepiece means a way through which we can see. Okay, there will be a hole through which we can see the two holes. In the two holes we will kept, in one hole we will keep the standard solution, and in the other hole we will keep the sample solution, and we will compare the color of both. The intensity of color in water is measured on an arbitrary scale. For public water supply, the number on cobalt scale should not exceed 20 and should be preferably less than 1 for public water supply. It should be less than 10 and it should not exceed 20. Okay. And for measurement of color, we should test the sample within 72 hours of its collection. So this is the instrument, this is tintometer and this is spectrophotometer. Both of the instruments uses this Nessler's tubes or some other tubes, and there will be provisions for placing this what is that, these tubes containing standard color and sample tube. So these are two instruments. There are many disadvantages of colored water. We have already seen some during discussion about the physical pollution. We have seen the disadvantages of colored water. Here are some. The colored water bodies are colloidal in nature, and it will absorb many metals and other things. That is one disadvantage. And this is unsuitable for certain industries like paper, laundering, textile, etc. Colored water is not suitable, and it restricts the penetration of light, and true color causing organic compounds except chlorine demand. So chlorine demand will be there. So these are the main disadvantages of colored water. Now we will move on to the second parameter, which is taste and odor. Taste and odor, odor is expressed as brackish, saline, salty, etc. So there are different types of odors: saline odor, salty odor, brackish odor, etc. Taste is expressed as disagreeable, earth, fishy, grassy, moldy, peaty, sweetish, etc. So this is the difference between taste and odor. Expressions are different. And if odor of chlorine or iodoform is found, it should be recorded. Chlorine or iodoform odor should be recorded. Okay. Uh, Mainly, this taste and odor is measured by means of an instrument known as osmoscope, which contains two tubes. Rented two tubes will be there, and we will smell both the tubes to find the change in color. So one tube is dipped in flask containing diluted water, and the other tube in flask containing standard water. So this diluted water is our sample, diluted in different what is that different milliliters of distilled water, different ratios of distilled water, and the other tube is our standard water. So we will compare the smells of both. Water to be tested is mixed with odor-free water, and the mixture at which odor becomes detectable is determined. So we'll mix the water with odor-free water, and we will see at which odor the smell becomes detectable. So that is the thing. And the number of times water is diluted determines the threshold number. There will be a number known as threshold number. So first of all, we will dilute this water. How many times we will dilute this water? That is known as the threshold number. For example, if 40 milliliter of sample is diluted to 200 milliliter, then we can say that the threshold number is 5. That is the thing. And for public water supply, the threshold number should be less than 3 or preferably 1. 1 is better, but it can be less than 3 also. Now the third parameter is temperature. You all know what is temperature. The desirable temperature of potable water is 10 degrees Celsius. It is very good. Below 10 degrees Celsius, it is very good. And when it is greater than 25 degrees Celsius, it is objectionable. We have to check the water, whether it is good for health or not. We have to check when it is 25 degrees Celsius or above. And when temperature increases, the multiplication of bacteria will be more rapid. When water above 15 degrees Celsius are to be cooled down as quickly as possible before doing analysis. So the temperature is a very important factor which causes the multiplication of bacteria. So that should be tested, and we will use the normal thermometers. Here we can see a figure. So this is a figure of a thermometer for measuring temperature. Density, viscosity, vapor pressure and surface tension are the other parameters which depends on this temperature. It also determines the rate of chemical, biochemical and biological activity in water. So these are the importance of temperature. Now we will see the next parameter. This is density. It is measured by using an instrument known as hydrometer. Hydrometer is nothing but it is a tube containing a, here we can see the figure of a hydrometer. So this is a tubular shape which contain a thin tube at the top, and at the bottom of the tube there is a broad surface below the bottom. So inside this broad bottom, there will be some what is that ballasting materials like mercury, lead, etc. That will give the stability to this bottle. Okay, it has a very curved end, so this liquid will give us stability to this material. I mean this tube. So these are the parts of a hydrometer. This hydrometer will be inserted in water. So what will happen, this hydrometer will will be floated in water, and if the water is not good for health, then what will happen, it will be immersed a little. Okay, so this hydrometer will be immersed a little, and the reading, there will be a graduated, this tube will be graduated, and this reading will be noted, the reading at which this uh, this portion of the hydrometer coincide with the water will be noted, and this will give the density, relative density of that particular water. So hydrometer is gently lowered and floated into a graduated cylinder containing the water sample. The point at which the surface of the liquid touches the stem of the hydrometer correlates the relative density. The next thing is electrical conductivity. Electrical conductivity is a parameter which shows the salinity of water, and it is measured by using EC meter or conductivity meter. It has actually two different cylindrical electrodes. So two electrodes will be there. The distance between we will keep a distance between these electrodes. When these electrodes are inserted in water, we will apply a voltage. So what will happen? The resistivity of water will be the resistivity of water between these two electrodes will be noted. So that will give the electrical conductivity. So resistivity is equal to 1 by electrical conductivity. So we'll get the electrical conductivity of this is an instrument. This is the EZ meter, which is digital. So this is the sensor. This may contain two electrodes. So this is the thing. And another important physical parameter is the radioactivity. It is measured by using solid state nuclear track detector. So this is an instrument which we will place over the sample of water, and this will give the amount of alpha rays or radon 222 which is released from the water. So the detector should remain undisturbed above the sample for at least 3 weeks. Then only we will get the reading. And another important parameter is viscosity. Viscosity is measured by using viscometer. We all know that viscosity is the resistance to flow, liquid's resistance to its flow, and it also describes the internal friction of a moving fluid. The viscosity is always inversely proportional to temperature. When temperature increases, viscosity decreases. So this is the relation. And there is an instrument known as viscometer which is used to measure the viscosity. So this figure shows a digital viscometer. So these are the things. Thank you.